Okay, now we're going to get into more of the nitty-gritty of the nervous system. And again, some of you, this is going to be so familiar. Feel free to skip through. You don't need to watch every little thing. You'll have the notes. But for those of you who actually want to kind of get some explanation, we're going to start on this particular aspect of the nervous system, talking first about that general layout. And there are the divisions, and the two basic divisions are the central nervous system, CNS. All of that system is located in the skull and in the spine. So that means it's the brain and the spinal cord. And keep in mind that those are bony structures that are taken care of, that the brain and the spinal cord are being protected by bony structures. The other division is the peripheral nervous system, or the PNS. And that is the part of the nervous system that lies outside of the skull and the spine. So that's a pretty easy way to kind of remember where you can locate each section. And there are two divisions of that PNS. Biopsych of stress students, we've already covered this. And so if you're taking this course as well, or this is a subsequent course from a different semester, this should seem very familiar. The somatic nervous system is that system that interacts with the external environment. And we have a couple of types of nerves. And I always like to refer back to the word affect and effect. And writing, lots of people mix those two terms up. It's like, oh, that's an affect. It's important to see that affect. This storm will affect this area or this city, okay? And affect is the result of that. So think of it that way. So, oh my gosh, the effect of this storm was to destroy 5 houses, okay? Now, let's use that same AFF and EFF prefix. Afferent nerves carry information to the CNS, to the brain and spinal cord. So here comes the storm, it's carrying information. Efferent nerves carry the result of that, well, I shouldn't say the result yet, carry that information to motor neurons and then the skeletal muscles, okay? So that's important. So the effect of those nerves that carried information to the CNS might be, you're going to go like this with your hand. You're going to jump back. You're going to take your hand away from a hot stove, okay? So the autonomic nervous system, or ANS, is the other division of the PNS. And this division regulates the internal environment. We need to get information from our internal environment, from our organs and tissues to our central nervous system, okay? So think about this as a loop within your own body, and it's not reaching out to the environment, the external environment. And here, think about afferent nerves. They need to affect the CNS, right? So afferent nerves carry information from those internal organs. We can even just say your stomach to your CNS. And then efferent nerves carry information from the CNS to internal organs. So you have this loop inside, within your body. Now, we've got two more divisions, two divisions of the autonomic nervous system. Again, biopsychostress people, this should be like so familiar by now. We have the sympathetic nervous system. Oh my gosh, I have so much sympathy for you. There is a bear and I'm part of you, so it's us. We got to get away from the bear. So I'm going to kick in and do everything I can to help you with me inside of you get away from this bear. Now, after you get away from the bear, you get back, as I talk about, in the cabin, and you bolt the door and you've got all your windows closed and big wooden bolts, and there's no way this bear is going to get you now. And this is assuming the bear even wants to get you. We'll talk about bears more at some point, I suppose. But so now your sympathetic nervous system no longer needs to be activated. It needs not to be engaged. Instead, that second division of your autonomic nervous system, which is your parasympathetic nervous system, kicks in. Now, let's get back to that more relaxed, normal state. 31 pairs of spinal nerves. We won't, in this particular class, be dealing much at all about the spinal nerves, but I certainly want you to be aware of that. Remember, 31 pairs, and then 12 pairs of cranial nerves. And even though I won't be talking a lot about cranial nerves, it's really important to understand that there are these pairs of cranial nerves, because when we talk about different structures in the CNS, we will say something about nerves are found, that the head of nerves are found in these areas. So if you look at it this way, we have a system of twos. Our nervous system is a system of twos. I should have rearranged it a bit, but here we go. And so here across is the first division, CNS and PNS. The CNS breaks into two divisions, the brain and the spinal cord. The PNS, remember, autonomic and somatic. Now, things get shifted sometimes on these, so I want to make sure you can see that you should draw lines, okay? Draw lines from PNS to autonomic, PNS to somatic. Now, let's jump to this autonomic, and that has two divisions, which are the parasympathetic and the sympathetic. Okay, so this guy's out by himself. And here is a more detailed a flow chart, if you will, of the structures. And you can see that basically I didn't go all the way down this whole flow chart diagram, but if you are interested in following through into more specific levels, this is available for you. Now, I already mentioned that the CNS is basically encompasses the most protected organs in the entire body. They're encased in the bone and covered by meninges, or one is a meninx. Again, one of my pet peeves are the way that our language kind of takes words and fools around with what the plural should be like. I would like one nice rule, not like one goose, two geese, one mouse, two mice. I can go on and on and on. And so meninx doesn't sound like one, but that's what it is. And these are protective membranes that are arranged in a very specific way. And the most outer, let's take your scalp, let's split it back. Don't worry, we can go and stitch it back. Nobody will know. Your hair will grow back, no issues whatsoever. Okay, now let's just cut a section of your skulls and lift that off so we can kind of look inside and start moving down. If you have brain surgery, guess what? They're doing this. So the outermost membrane that we will encounter is the dura mater. Literally means tough mother. And it is tough. It has to be like the one that is going to be able to provide protection from outer skull, different things. And in a minute, I'll go back and kind of go through this with a little bit of an analogy, but let's keep moving down. So we get the dura mater. Let's just move that, cut it a little, move it out of the way. Now we keep going in towards your brain and we get to the arachnoid membrane. Arachnoid, spider, arachnophobia, does that sound familiar? And it is more of a kind of like a spider webby type of membrane. So it isn't that tough. I'm gonna like a balloon rubber tough membrane like dura mater. And underneath that is a subarachnoid space. That's easy to remember, below the arachnoid. And cerebral spinal fluid flows through this. It is also a site of some brain bleeds and trauma. And I'll come back and refer to that again in a few minutes. And then we go all the way inside and just gently wrapping around your brain matter itself is the pia mater, the innermost membrane. And pia mater literally means gentle mother. We want that gentle mother membrane to be the one that is in contact with your brain itself. And I kind of use the analogy, if you wanted to take this very this very sensitive, fragile, and very important item and ship it across the country, excuse me, I get the hiccups. You would not just pick it up, put it in a big old cardboard box, put your label on, put it in the mail, watch the mailman pick it up and toss it, not that he would, but let's just pretend, toss it into the truck and believe it's going to be okay when it reaches the other side. If it contains liquid, that's even more. Here's a very fragile, delicate item and it's filled with some liquid, okay? No, we wouldn't do that. First, we would take this a precious item, your brain, now think of it this way, we would take that and we would wrap it with something, some very soft tissue paper, something that would give it some protection, but it isn't real tough. And it's a little more like able to be ripped than other things that you might put in, but you're wrapping it with that. You might even take a real thin piece of nylon. Let's do that. Pia mater, okay? Now, I put it in the box and I'm like, wow, this thing can like be jungled all over the place. It can be moving, it can be bumping. I would like some of those really cool styrofoam chips or bubble wrap. Bubble wrap is really good for getting rid of frustration. You can pop those little bubbles in and you're not really hurting anything unless you want to reuse it. And so, but I'm like, oh, wow, I'm just gonna, you know, maybe I'll do the bubble wrap. I feel better and I'm really gonna stuff it around. That would be like the arachnoid membrane. And then finally, the outermost dura mater, I might put in some of that, you know, like the brown paper, almost like butcher paper that sometimes is crammed in to your packages. I know we all have done a lot of online shopping, at least most of us have. And then, so that would be, we could wrap all that within there, but the outermost, and let's call it your skull is going to be the cardboard box that you put it all in. I've changed that up a little bit from previous years, so I like this way a little bit better. So hopefully you can see that there's a reason for the ordering of these particular membranes. Now, I mentioned cerebral spinal fluid, okay, CSF, and that fills that subarachnoid space. It goes down the central canal of the spinal cord all the way through and through the ventricles of the brain. The ventricles are hollow until you fill them with CSF. And we'll talk a little bit more about that in a minute. But it also, this fluid helps to support and cushion the brain. And it's produced by something called choroid plexuses. And again, I'd like to call this choroid plexi. Whoever invented the plural, not sure. If you block this cerebrospinal fluid somewhere in your CNS, it can cause hydroencephalus, water on the brain. And some of you have heard about that before. So here's a little illustration. Here are the 4 ventricles. There's 4 ventricles. There's this kind of duct that carries it down through to this area. And then if you look, you can see this central canal move all the way, if the picture continued, down the spinal cord. I want to mention something about these meninges right here, because lots of times people just think about them as being around the brain and somehow right here they stop. No, these meninges go all the way down around your spinal cord. So they are continuous. If you have a spinal anesthetic or spinal tap or something, they are actually going to insert a needle. It will go through the membranes surrounding your spinal cord and puncture them. Generally, this is not a problem. I can tell you that this system, by experience, that's why I have a lot of weird experiences, and I found a way to weave a lot of them into my coursework. So I'm like, okay, it sucked, but now I can talk about it in this way. I had to have some abdominal surgery at one point, and I was like, I don't want anesthesia. I don't want general anesthesia. No, I might not ever come out of blah, blah, blah. Anyway, so I talked the anesthesiologist into just giving me a spinal to numb my abdominal area, and that meant injecting anesthesia into that area of my spine, and give me something just to make me feel like time is going, you know, like la la la, which they did, by the way. I really had no concept of the passage of time. The only thing in this was this anesthesiologist, and it might have been he should have retired a while ago, I don't know. But he came in, I'm laying there on the table, and he starts to give the first spinal injection. It should have only been one. And I hear, oh, and I feel the needle come out, and then I feel it puncturing again. And I hear this little utterance. If you're going to be a doctor, any of you, don't go, uh-oh, ever, when you're working on a patient, ever. Or, oops, oh, not good. Or even go, like that. That means you're upset because something just happened and didn't go well. Anyway, 4 times. Okay, no deal. Surgery went fine. First time I tried to sit up, they go, okay, sit up now. You need to start drinking water. I got the mother of all headaches. I can't even describe how bad this headache was. And I'm talking about that because this balance is so critical that if you disrupt it, you can end up with a headache like you've never experienced before. And I got sick. I don't like telling you guys this, but I vomited like a lot. So this happened for 24 hours. They're trying to get me past this. The next, after like 36 hours or so of this, horrific experience, my doctor, who actually did the surgery and did a fine job, by the way, came back in. They took me back into surgery, ran a line from my own blood, from my own vein, into my spinal cord back where the injections were done. And what they were doing was a blood patch. this is something that gets done. It's called a blood patch. They use your own blood for a lot of obvious reasons. It's right there. You don't need much. And it was so immediate. The relief was so immediate that it was uncanny. And I'm thinking, and why did you not do this like 24 hours ago, maybe? But that blood helped to patch the holes in those membranes. And now my cerebral spinal fluid balance was appropriate. Now, I just like to tell that story because not only does it tell you like if something like this happens, what you should request be done, and not that it happens very frequently, but it is possible. I'm sure I'm not the only one, but also it shows the incredibly intricate workings of our physiology. Absolutely, I mean, it's magical. So anyway, excuse these bigger fonts on this particular, your partial outlines, I have reformatted and gotten them all in a smaller font. I just was in a big hurry to try to get this up for you, so please forgive the differences. The blood-brain barrier. We have a barrier, okay, that surrounds the brain and our CNS, and this blood-brain barrier helps maintain a very precise electrical balance, and it also helps to keep out toxins. This is really important. We don't want our brain just to be like, oh, let everything in, I don't care, you're toxic, oh, that's okay, welcome. No, not in our brains. How is it that we have this barrier? Okay, in our blood vessel walls, in the rest of our system, we have little tiny gaps between each of the cells, so that fluid can be extruded through those vessel walls, okay? And we need it like that for a lot of functions, for getting nutrients and things into certain areas, available to tissues and structures, and also to pick up waste and to carry those away. However, we're going to protect our brain with this tightly packed cells. They are that's called tight junctions. And because they're put together so tightly, you're not going to have that movement in and out like we might somewhere else in our body. And what does it keep out? Big proteins and other large molecules that make up a lot of the things we don't want in our brain. However, it has little pumps that make sure that we get glucose into the brain, because your brain is incredibly metabolically active. It will preferentially grab glucose as long as it is able to if you are low in glucose. Now, fat-soluble molecules can get across. The lipid aspect of them allows them to move across, and this includes sex hormones and also some drugs. Now, there are other drugs that they're water soluble, they're not going to be able to get across. And when we talk about some of the things like treatment for schizophrenia, we're going to talk about that, or I should say treatment for Parkinson's. That's a better selection. There's reasons why we can't use certain neurotransmitters and just inject it and expect it to go into our brain where we might want it. That is down the road. Hopefully you'll still be hanging out with me at that point in time. So this is just an illustration of kind of a section cut across of the blood-brain barrier. And you can take a look at that, and hopefully that will help you out a little bit. So now, cells of the nervous system. I didn't used to even address this, and then I started finding out that there actually were students who just didn't have anybody spend much time in 101 on these things. And I want to make sure everybody has all the information they need as we move on to the more complex aspects of this course. Neurons. These are cells that are specialized for receiving and sending electrochemical signals. And we have external parts. I'm going to go through. This is the illustration. You can follow along on your partial outline if you want, because I did minimize that font and I could fit it in everywhere. But make sure you get all of these listed. We have the cell body. We'll talk about what that's important for later. Dendrites, axon, the axon hillock, terminal buttons, myelin sheath, nodes of Ranvier, it's a French word, it's not Ranvier, it's Ranvier, and synapse, and the cell membrane. Okay, we can look at this. You've looked at all of these. The cell body is this section right here. It houses, I'm going to talk about that more later. And this is the axon, and this is the axon hillock right here. Here are the terminals. These spaces at the end of the terminals, are those, they're very small spaces, are the synapses. This is myelin sheath. It's a lipid substance. We're going to look at that more closely in a bit. I just wanted to make sure you had all of these terms of the external features of a typical neuron. Now, this is a textbook neuron. Can there be differences? Of course there can be, but they all need to have these particular features to operate the way they need to in one form or another. So now let's go through the internal parts. The nucleus. The nucleus is found in the cell body. Mitochondria are found in a couple of different places, and we'll talk about that. People very often think about it only as being in the cell body area of the neuron, but they are also, they're like the little batteries, like the energizers, if you will. They're also found down in the terminal endings or buttons of the neuron. Cytoplasm is that fluid within the cell, cytocell plasm, that fluid. Endoplasmic reticulum. These are, there's two forms. We find these in the cell bodies. And there's two forms, smooth. The smooth ones are important for forming fat, which we need. Oh my gosh, we need fat so much for our brains, for cell walls throughout our body. And so this is important. There are some areas of endoplasmic reticulum that are rough. They have, it looks like they have little beads on them. And those are ribosomes. And the ribosomes are important for protein synthesis. Then we have the Golgi complex, microtubules, and here we go. Here's an illustration of all of those different parts, the internal parts of our neuron. And you can see here that we have the nucleus, okay? We have the mitochondria. We have the endoplasmic reticulum, the smooth ones, again, for fat synthesis, and the rough ones where there's little bumples, and it's really hard for me to see on my screen right now, but if you see little bumps on them, that's the rough and important for protein synthesis, and those little bumps are ribosomes. This here is the Golgi complex. That is important for taking, packaging, and moving products from the nucleus and that cell down to other areas of the neuron. And these tubes are, guess what? The microtubules. Now, I'm going to finish this part of this lecture with the cell membrane. And then I'll pick up with a part 2. But the cell membrane first is a bilayer, 2 layers, and a phospholipid bilayer. So that means it has, what is this? This should look familiar. This should look familiar, I'm hoping, the fats. And we actually have 2 layers of the fat and molecules, phospholipids. I should have put that all in there. And protein molecules are embedded in these sheets. And again, at the very beginning of the next lecture, I'm going to come back to this first, go through this material, and give a little demonstration of what this bilayer is. It's so important, the aspects of it. So I'm going to come back and pay special attention to it. These protein molecules are either channel proteins or signal proteins. And when I start talking about this again, I will be more specific about what is going on with the channel protein and what is going on with the signal proteins. They operate very differently. Now, in these sheets, these 2 layers, there are section that's going to turn inward because they're the tails and they're hydrophobic. Well, what's outside of the cell and what's inside the cell? Fluid. These tails, and I'll show you the picture in a moment, are like, I don't like fluid, keep me away. So they turn in to face away from the fluid. And they are non-polar. However, there are heads of these molecules that go, I love water, I love it, I love it, I love it. Let me be next to fluid. Let me be next to the cytoplasm or extracellular fluid. And these are polar. And here's that little illustration. And here, let's go through and see. Here is one of the layers, okay? And down below is the other part, the other layer, and the tail's facing inward, the head's facing outward. So we have extracellular fluid on this side and intracellular fluid on this side. And this is a stable arrangement that keeps these 2 layers together. Otherwise, our cells would be falling apart within us. Here is a channel protein right here, and you can see that things can flow through. It's basically a receptor site, and when we get to neurophysiology, we will need to know this and come back and talk very specifically about that. This other, look at this little loopy thing. These are signal proteins, and you can see they connect from the outside to the inside as well. However, they loop, they loop, they loop, they loop, they loop, they loop. And you're like, oh, this is different. Just a hint, gonna take longer. Interacting with this is going to cause an effect, but it's going to take longer for that effect to occur. And geez, I wonder if there can be more than one type of effect. A little hint. And wrapping up just with these particular illustrations, classes of neurons, multipolar. Here is it. You see the multipolar. It's very common with a motor neuron. And here are dendrites. You can see where the cell body is, the axon, the terminals. Unipolar, look at the one that's labeled unipolar. Here, it really has the cell body like stuck over here. It has one process coming from it. That's where the unit comes from. And then that can split both ways. That's very common, typical of a sensory neuron. Bipolar, we're not really going to talk about pyramidal cells right now. Bipolar, here we've got the cell body here. We have one process going this way, one process going this way, bi. And those are used a lot to take information from one neuron to another neuron without being necessarily physiologically involved in an effect. So we call these interneurons. Special definitions you need to know, and I rest my case again about, go away here, you, about plural. If you're in the central nervous system and you have a cluster, a group of cell bodies, a cluster of cell bodies all together, okay, in the CNS, those are referred to as nuclei. One cluster would be a nucleus. It is not the same thing as a nucleus within the cell. Wish somebody would have been a little more creative when they named all these things, but I wasn't alive then. And if you go to the PNS and you have clusters of cell bodies, again, they're now called ganglia, those clusters. One cluster would be a ganglion. And then for, I have this little thing down below that's driving me crazy, but bundles of axons. If you have a bunch of cell bodies and they have axons, they're going to be a bundle streaming away from that nucleus, or several bundles would be coming away from nuclei, and those in the CNS would be called tracts. In the PNS, they're called nerves, okay? So I want to stop right there, and again, give you this much to start with, and then I'm going to come back and continue with these same outline notes, but going back and discussing that cell membrane again for being a little more descriptive, using analogy so that we have that down. Again, those of you who've had this like a million jillion times, or have taken like cellular biology, microbiology, all that, I am sorry if this is like, oh my gosh, skip through it. It's fine. You already know this stuff. You'll know what you need to know at the end. I don't mean to be tedious, but I know there are many, many of you who, this sounds familiar, but honestly, I don't remember. Okay, and hopefully going through listing these things and then describing them is helpful. So, hey, hold on to that thought. I'll see you back again in the next section of this note lecture, if you will, video lecture, and hope you can regroup.